Okay, I think let's resume. And now we are going to switch gear a little towards cancer. So as I started talking about phylogenetic analysis in general, now we are going to focus more on how to do it from tumor sequencing datasets. Okay. So as you probably all know that somatic mutations are causing cancer, right? We have a healthy genome which is acquiring some sort of somatic mutations, which is giving the cell some sort of selective advantage, proliferation advantage, and then that basically leads to uncontrolled cell growth coupled with, let's say, accumulation of more and more somatic mutations, which is finally creating this tumor mass. Okay. So the question is, this somatic mutation process, can we really think of it from the perspective of evolution? So it turns out that in 1976, Peter Noel came up with this theory of clonal evolution. So where basically he was saying that there are multiple tumor subpopulations which are arising over time, and cancer is basically a hotspot of tumor mutation accumulation and selection process. So what we see over here is that x-axis represent time in terms of cell division. So we have a bunch of cells, these are in homeostasis, and suddenly there is a particular cell which is turned rogue by acquiring certain mutations, and then it basically starts proliferating much faster as compared to other cells, and then starts accumulating more and more mutations to create the tumor mass. So let's say we take the data at this particular time point, right? This is our snapshot data. Let's say we have performed some sort of sequencing to obtain the data set. And what we can see is the presence of multiple subpopulations within the same tumor, right? And this is known as intratumor heterogeneity, and which Mohit was referring it to earlier, where we saw that, yes, maybe through a targeted therapy, cancer basically goes down for some time, but that it again relapses. The reason is that maybe there are some dormant populations which are again becoming more and more active after the therapy, right? So that's why it's not only good to understand just the dominant population, but maybe all the populations. That's when we can design targeted therapy for corresponding to all different populations, right? So we want to study more of this clonal evolution process because, as I said, this intratumor heterogeneity is actually very disruptive in nature, in the sense that it can contribute to so many tumor phenotypes, including drug resistance. It can cause relapse, it can cause phenotype changes, plasticity, and so on, right? Everything. So this heterogeneity is basically underlying a lot of cancer phenotypes. Question is, how do we characterize this kind of heterogeneity in cancer? And can we characterize the evolution process a little better? So that is what are the interesting questions to me, which I started looking at during my PhD time. So what are the aspects of cancer that we are going to discuss today are this set of questions: that what mutations are present in the tumor sample, okay? In what particular order these mutations actually arise, and what can we really characterize these cell subpopulations within the tumor, and maybe this characterization can be in terms of the mutations, okay? So essentially, if we represent a heterogeneous tumor sample, something like this, where these different colored shapes are representing different clonal or subclonal populations, then we want to really answer this kind of questions: that what are the composition of each of the clones in terms of the mutations, and what are their fractions in the whole tumor tissue, and is it possible to really recover their phylogenetic tree, that is the evolutionary tree structure, which connect all these different samples or different subpopulations, right? So these are the underlying questions, these are the interesting questions that we want to answer. What kind of data to utilize? Well, there are a lot of different sequencing approaches possible. The more traditional kind of approaches are these deep sequencing approaches or bulk sequencing approaches. So you have a heterogeneous tissue, different colors representing different subpopulations. What you do, you take a tissue sample, you sequence it at very high depth. What you end up with are basically this kind of variant allele frequencies, for the different mutations. So what you see over here, these are different mutations, and maybe they are corresponding to different subpopulations, but we do not know what exactly they are corresponding to, because the taxa of the phylogenetic tree or the subpopulations are not really well defined with respect to this. There is another set of techniques where you can perform multi-region sequencing. So instead of taking a single sample from the whole tissue, maybe you can take different samples from different regions of the tissue, right? And then what you end up with that is also like different views of the whole tissue, right? So multi-region sequencing is something, some extension of deep sequencing approach, you can say. And the third type of data that we can utilize, which is more novel in terms of the chronology, is single cell sequencing. So what we are again talking over here is single cell DNA sequencing, where you can really get sequencing data corresponding to individual cells, and you can probably measure the mutations in individual cells. Okay, so this particular approach became more popular, let's say 5-6 years back, and there have been a lot of developments in this particular space, and it has been applied in answering so many different types of questions related to cancer, whether it's of course studying of the intratumor heterogeneity and clonal evolution, but also looking into more circulating tumor cells, how metastasis, the migration pattern of the cells and response of the cells to different therapy, and so on, right? So we are going to address it, or we are going to look at this particular question of tumor evolution from single cell sequencing datasets. So in order to go there, let us first try to understand single cell data a little bit better. What is exactly happening? Okay, this is again an illustration of the heterogeneous tissue. You isolate the cells, you extract the DNA. Now what is happening is basically you are ending up with picograms of DNA, which is not enough to prepare your library for sequencing. So that's why these cells, or the DNA from these cells, they have to go through the steps known as whole genome amplification. So you are replicating the you are replicating the available DNA material through different techniques. There are multiple different techniques for whole genome amplification to give you enough amount of DNA so that you can prepare your sequencing library. Okay. After that, what you are doing, you have individual sequencing reads corresponding to individual cells. You are aligning them back to the reference genome, and that gives you sequencing data, right? Aligned sequencing data. And then you can perform different types of analysis. You can perform mutation calling, you can perform tumor phylogeny analysis, clustering, whatever you want. Now, one such question, one very basic question that arises that, well, we have sequencing data from single cells. Now, back in the day of, let's say, when bulk sequencing was more popular, people have already developed, at least mutation calling algorithms have already been developed for bulk sequencing data. Can't we apply the same techniques? Turns out, yes, you can, but then you will be losing out on a lot of things. Why? Because when the single cell DNA sequencing process is being introduced to generate this kind of data, there are some inherent challenges with the biotechnological assays, which we need to address. Otherwise, what will happen is that we are going to end up with a lot of false positives in our data, false negatives in our data, and so on. So let us first try to understand what are the different data specific challenges, okay? So this is a particular figure where we are comparing bulk sequencing against single cell sequencing. So what we see here, let's actually talk about that. We have bulk sequencing data. Again, we have the reference genome, there are different reads. What you see here is uniform coverage across all the different positions. What do I mean by coverage? Coverage is nothing but number of bases that you see at a particular position. So we know that in sequencing data, there are a lot of redundancy, and this is giving us statistical advantage in the sense that we have supports for the event by multiple reads, right? So that means almost every position is covered very well. These are again genomic positions I'm talking about. And this coverage uniformity helps us in identifying mutations. Now, what happens in case of single cell sequencing? Okay, there are a lot of different things. And all these artifacts that I'm going to talk about, these are artifacts associated with the whole genome amplification process itself. So what is the most prominent kind of artifact is something known as allele dropout, or ADO in short. What is that? So let's say this is a particular cell. Here you see a particular mutation. Okay, the star is representing a mutation. So we call it a true SNV. Okay, the star is basically a true SNV. But you look at the data and what is happening, let's say at this particular region, there is actually dropout of a particular allele. So you see that we have two alleles, right, at this location. One is mutated, the other one is non-mutated. Now, in this particular scenario, it happened in a manner, the sequencing during the amplification process, only the mutated allele was amplified, but the other allele was not amplified. So in this example, it is like this. It can happen the other way around also, right? Maybe the reference allele is amplified, but the mutated allele is not amplified. So then we lose out on the information about the mutation. So this is allelic dropout. Second thing is, again, allelic imbalance. Again, it's kind of similar to allelic dropout. In the first case, there was complete dropout of one allele, but in the next case, it's basically some amount of imbalance. So if the mutated allele is amplified, there is no problem. There is still some amount of problem, but it's basically stochastic. You don't know whether the mutated allele will be amplified or the reference allele will be amplified. So if the mutated allele becomes dropped out, then you are losing the mutation. So ideally, yes, you should see a variant allele frequency similar to 50%, right? Now the third thing is basically the locus dropout, which is nothing but a missing data. You don't have coverage at all. Let's say coverage at a particular region is 0, and if there was a mutation there, you will completely lose out on that, okay? And the fourth thing is something we call false positive error, okay? What you see suddenly in the sequencing data, you see the signature of a mutation, okay? And there is a signal that, okay, this is a mutated allele, but actually there is no mutation in the true genotype. Why this is happening? Because the enzyme which is being utilized for single cell sequencing library preparation, it can make some mistake. And because this kind of amplification is happening on the copies being made, the error, if that happens very early in the process, it will propagate down to other reads, okay? So this is something that we are calling as false positive error, because it will still look like a mutation, but it is nothing but an artifact, okay? So all these different kind of errors can be present in your data. Now the question is, given all these things, which were not a problem in case of bulk sequencing data, how can we still recover, let's say, tumor phylogeny or even the mutations? Let's say if we talk about the mutations, how do we distinguish the true mutations from this kind of artifacts? That becomes our interesting question to begin with. Okay.
So what are you missing in bulk sequencing?
So
just tiny clones?
Not just tiny clones, but let's say with bulk sequencing data, you are getting an average signal where the multiple clones are mixed together. It becomes a difficult problem to, let's say, separate the clones also. Why? Because first of all, in order to separate the clones, what people are dealing with or people are using is this signal in the VAF. So they are saying, okay, there are 60% alleles or 60% of the reads that are mutated, and we are going to sort of look at just the VAF, and we are going to cluster the mutations. Now, that is not really true, because VAF is a complex function of, let's say, tumor cell fraction, what fraction of the cells are actually mutated, and all these things. So what we are introducing is a lot more uncertainty, or the parameters that we are introducing, sometimes this is just an average. It's not really the true data, and we have to make a lot of assumption to say anything meaningful. let's say. No, but
I'm just trying to understand what's the real advantage of single cell other than.
So the advantage is,
Do it at all.
I'll tell you what the advantage is. Again, the methodology that I just described, yes, they are not depending on, they are actually treating the sites as independent. But even within the same site, if we are not able to say, okay, if this is a particular clone, like what are the combination of mutations in a particular clone? We are not able to say that with bulk data. We can only say that, okay, this set of mutations, they have a similar VF, so as if they belong to the same clone, but that may not be true.
From what I understand, if you have two different clones within the same population, they might have very different mutations, right?
Yes, they might have very different mutations. So let's say, just to give you a simple example, okay? Yes, so let's say these are the two clones, okay? These are separate set of mutations. Let's say we call it M1, M2, M3. So let's say these are two clones. This is C1 and this is C2. These are only two clones where M1 is only present in C1, M3 is only present in C2, N2 are common across both the things. If you look at the bulk data, you will probably say, okay, this is one clone, this is another clone, this is another clone. But ultimately, there are actually only two clones here. And the things become more complex when you have more number of clones, right? Yes, in most cases, you use paired end reads. Not always. Okay. But certainly, you can use that as some sort of signal. Okay. So again, coming back to this set of problems, so we have been developing methods for this kind of single cell sequencing data sets. And earlier methods, actually we looked at set of methods, or we actually developed set of methods where we started from observed genotype of mutations. So what do I mean by that? That you have the sequencing data, you have applied some sort of mutation calling algorithm, and that has already given you this kind of profile. So these are somatic mutation profiles for individual cells. Your columns are representing cells, your rows are representing mutations, and you have a binary kind of matrix where you, one basically says that mutation is present in that cell, 0 means it's absent in that cell, and in some cases you have some dashes where you basically have no data, right? So given this kind of genotype matrix, you can actually develop phylogeny reconstruction method. So we developed one such method back in 2017. It's called SciFit. And then it was extended to infer the cancer populations also. So that method was published in 2019. So one of the problems with starting from this kind of observed genotype data is that, first of all, you are adding another level of uncertainty in the sense that you are relying on a variant caller to give you this kind of things. Secondly, all these methods that I'm talking about, they work well with small size data sets. What do I mean by small is, first of all, they are small in terms of number of sites, and they are also sort of small in number of cells that you can really handle, okay? So later on, we have tried to develop methods where we can directly go from read count data to this kind of phylogeny as well as mutations. So essentially, instead of treating this as a two-step problem where we first find the mutations and then we actually use the mutation data to sort of look at the tumor phylogeny, what we are saying is that these two things are related. Ultimately, if there is an underlying phylogenetic structure that can help us get this mutation better. So if we jointly analyze this problem, if we jointly solve the mutation calling and the phylogeny inference problem directly from the observed read counts, then probably we'll be in a better shape, right? And using this kind of approaches, we can even make more, I would say, scalable models. Scalable in terms of number of sites, which you see over here, and scalable in terms of number of cells as well, which I'm going to describe a little bit of this method as well. And this is still work in progress in some sense. So we'll start with the first method. And before that, I'm going to discuss a little bit more about the tumor phylogeny inference problem. So now, if you think about the single cell sequencing data, it's actually very ideal for phylogeny inference. Why do I say that? Because in the ideal scenario, if you have a matrix like this is very simple character matrix, 1 and 0. These are the possible values. So far I have talked to you about all these fancy methods where you are looking at DNA sequencing data sets, AC, TG, those are the possible characters. Here you have only two things, 1 and 0. So simple, you can just apply distance-based methods and you can get the tumor phylogeny, right? Now, where is the problem? The problem is that it is not ideal. Your observed data has a lot of noises. You have false positive errors, false negative errors due to allelic dropout, and then you have some missing data. Now, with this kind of scenario or with this kind of data sets, how do you really ensure that your phylogeny inference is robust, that you are not making mistakes because of these artifacts? Now, with those things in mind, there was another line of methods. So this is actually a paper from 2019. So where they showed that indeed the phylogeny structure can help in calling mutations. So I would say that this is probably one of the first works which tried to treat the problem in a joint manner, that instead of solving mutation calling and phylogenetic inference as two separate problems, why don't we do it together? Okay, so that's what they did. So they start with this set of sequencing reads from individual cells. They will try to reconstruct a tree, and from this reconstructed tree, they can try to find these mutations, okay? Mutations in individual cells. So essentially, at the end, they are getting both these things. They are getting a phylogenetic tree, which explains the data, and they are also getting a set of mutations present in individual cells, okay? So this method is called Sci-Fi, and it came from a lab in ETH Zurich, okay? So what they are doing, they are essentially assuming this thing that we have a bunch of somatic cells, and we know that they should be related via a phylogenetic tree. Why? Because ultimately cells are there because of the cell division process, which are again, we know should be coming from a binary tree, right? So somehow, if we are able to, and of course we have this mutation data, so we have the somatic mutation profiles. So these mutations are nothing but these are just propagated along the tree branches. So the tree structure is basically giving us more, I would say, more support for a mutation. It is basically saying that if there are two cells with very similar mutation profile, then they should be together in the tree. And at the same time, it will give us, I would say, more support in terms of distinguishing the mutations from artifacts, right? So that's what they utilized. So now, this was back in 2019, and what became problematic for this method is that there were new sequencing technologies that could scale up to very large number of sites. So what I'm talking about is single cell whole genome sequencing datasets. Okay, so that particular method that I just described, it was still valid for small size datasets. Okay, but as soon as you go on to work with, let's say, this kind of single cell whole genome sequencing datasets. So this is just one example. Again, this is from 2015, which was used in case of neurons. So they sequenced individual neurons, and ultimately they actually covered a lot of percentage of the genome in these individual neurons. And they showed that the method actually, this was a technological assay. So this particular assay, using this assay, they were able to get single cell whole genome sequencing datasets. And then around 2020 or so, this similar kind of technologies were being applied to look at cancer as well, okay? So people even got single cell whole genome sequencing datasets from different cancer patients. So with that in mind, what we observed that the existing methods, they cannot handle very large number of sites. Whether it's single cell whole genome sequencing data or whole exome sequencing data, either they would run into this problem of very long running time. So methods can run for 4 days, 5 days, and still no good result, okay? Or they can consume a large amount of memory. And in fact, so after sci-fi, we actually worked on a integer linear programming algorithm to develop a more combinatorial solution for the same. But even that actually led to a lot of memory issues. So with large number of sites or large data sets, it was not able to perform well. So to that end, to solve this problem, we wanted to develop a scalable phylogeny-aware variant caller. So essentially, what we want to do is we want to solve these two problems together. That okay, I have this kind of read count data coming from individual cells. Number of cells can still be small, but for each cell, I have a very large number of sites, which can go even up to millions. Okay, what do we do? How do we deal with this variant calling, this joint problem, variant calling as well as phylogeny inference? So if we are talking about phylogeny, and one thing that is for sure is that we cannot directly borrow stuff from species phylogeny, right? What do I mean by borrowing stuff? Again, I'm talking about model of evolution. Why? Because those are, again, models developed with respect to sequence evolution over time, where we are looking at evolution of genes and so on. Here we are looking at a very different evolutionary process, somatic evolution process, which is much more on a shorter scale, where the mutation rates are different, and probably the exact same models may not be applicable. But still we wanted to start with something very simple, okay? So this is exactly the overview of the phylovar model. What we do, we developed a likelihood-based method. So that means we have to come up with a model of evolution. And the model of evolution we resorted to is something called infinite sites model. And why we chose this infinite sites model? First reason is that it's a simple model. What it is saying that one site can mutate only once during the evolutionary course. Now, this may or may not be applicable in case of cancer, but we'll see that this is still a reasonable model. And what has been shown in literature is that probably around that point also, till that point, the infinite sites model actually was being applied to study cancer evolution in multiple studies, okay? So that is our model of evolution. Now, we need some other things. What are some other things? First of all, there is this simple filtering that we have to do. We have to first identify the set of loci for which we really want to study, okay? And we adopted a likelihood ratio test. Again, this is very simple likelihood ratio test, which would actually look at the available or the observed sequencing data, and it will determine whether there is a mutation there or not, okay? Yes? Yes. If, of course, it still shrinks, but still it's kind of big because let's say when you are talking about whole genome sequencing data, or let's say even if we talk about whole exome sequencing data, even after choosing this candidate loci, the other methods actually fail. Okay, so you may come down to something like 1,000, which are mutated sites, but maybe the existing methods were only able to handle 100 sites. That's what the reality was. So the third thing is that we also need a model for modeling this errors associated with single cell data, right? So we are going to talk about that also. And then we have to have a search algorithm. Now this search algorithm is really something that we can borrow from species phylogeny literature. We did, but with some added things. And that's what we are going to see, what we actually did. So we'll start with this model of tumor phylogeny inference. And as I said, this was being applied by other groups when they were talking about tumor evolution. And this model of evolution is basically infinite sites model. So what it is saying that each site or each mutation site gets mutated at most once, and mutations are never lost. So because we are looking at a much shorter time frame, what we are saying is that once the mutation occurs, it is going to persist. It is not going to get reversed or anything, okay? Or it is not going to get lost. So this is the infinite sites model. Again, infinite sites model has been utilized in species phylogeny also, but this is a very simple model. So it can restrict our search space, and it was being applied in tumor phylogeny inference by other methods. So we wanted to start somewhere simple. So this is something that we adopted. Okay. Then we have this tree model. So what is exactly this tree model? A tree, we have this tree topology consisting of set of nodes and set of edges. Okay. And along with this tree topology, we also have to have this mutation placement vector. So what is this mutation placement vector? Let me just draw it on the board so that it becomes easier. So let's say we are talking about this phylogenetic tree, okay? And we are talking about one particular mutation, M1. So what we are saying is that we have to follow this infinite site model of evolution, okay? So that means this M1 can be placed on this tree only once. So either we can place it over here, or here, or here, or here, like whatever is the number of branches, on one of the branches, M1 can be placed. So depending on where exactly M1 is placed, you are getting different mutation placement positions. So this is corresponding to M1. Similarly, you can have it for all the other mutations as well, right? So if J is representing the index of the mutation, and if we are looking at n candidate sites or m candidate sites, whatever it is, then we have to have this mutation placements ready for all the different mutations, right? Anywhere on the tree. So we are considering all possible positions of the tree, because out of all the possible positions, one of them will have the optimal likelihood.
I'm wondering, I'm asking because let's say in an extreme case, the mutation was present in all the cells. So then you know
it has to be on the root. Yeah.
Then if you place it at different places, it's suboptimum. Can you use that?
So it's very simple. You just associate one normal, and basically here it's all zeros, right? So then you can place it over here, and we can say this is a truncal mutation or a clonal mutation.
So that's what I'm saying. Do you use the information of frequency of the mutations to sort of bias where the mutation will go on that tree?
Not exactly the frequency of the mutations, but rather we look at the overall likelihood itself. So depending on where exactly you are placing the mutation, your likelihood function will change, and then the optimal placement you are getting by looking at the optimal likelihood. Okay. So this is exactly the representation of that. If we are saying that mj of q is equal to 1, then that means the mutation occurred at particular node q during the evolutionary history of locus j. Okay. So j is basically indexing the locus itself, and wherever it is happening, this is the node. Okay. So node basically saying if something is happening on the branch, you can also denote it as the mutation happening on this internal node. So the branch which is incident upon the internal node, we can use that also, and that is what is being utilized over here. Okay. And of course, as I said earlier, that at most one element in mj is allowed to be 1. So this is coming from the infinite sites model. Okay. So now we actually come to this single cell genotype error model. What exactly is this one? So as I said earlier that we have allelic dropout or the missing data, right, which can result in a loss of mutation information. So essentially there was a mutation originally in the cell, but in the sequencing data we are not seeing any signal because of allelic dropout, right? So that can give rise to something called false negative. On the other hand, we can have something as false positive. So what is the false positive? Again, you saw it earlier that in the sequencing data, we are seeing the signal of a mutation, but this is actually just an artifact of the sequencing amplification process, right? So to deal with those, we can just use this false positive rate. So we are introducing two parameters, one for false positive, the other one for false negative. Let's call them alpha and beta. And what are these? These are simple beta distributed numbers, okay? Why? Because ultimately alpha and beta are representing the probability of a particular site being due to false positive or false negative. So we know that probabilities can be modeled using beta distribution. So these are basically numbers between 0 and 1. Okay. We need two other things. So we need these two things. This is the true genotype, Gij. So this is the true genotype of, let's say, ith position at jth cell. Okay. And similarly, the psi ij, this is the library genotype after the whole genome amplification process. So the true genotype is something that is actually present in the cell. Psi ij is again a genotype which can be inferred, let's say, after whole genome amplification. So that means this is already containing some errors, but this is the true genotype. So in terms of these four quantities, I can define this probability distribution. What is this probability distribution? This is very simple. So if I am saying that true genotype is 0, that means the actual site is not mutated in that cell, right? True genotype is 0. And if we are saying that psi ij, that is the library genotype after whole genome amplification, is 1, that means this is a clear case of false positive, right? So that conditional probability will become alpha. Otherwise, it is 1 minus alpha. So it's a simple Bernoulli distribution that we are looking at. And in the other case, if we are saying that the true genotype is 1, that is the cell at that particular position contains a mutation, and if we are saying that psi ij is 0 or 1, then if psi ij is 0 and gij is 1, then we can model it using beta, because what is beta? Beta is just the false negative probability, right? So given these two scenario, given these two values of these variables, the conditional probability will be beta, otherwise it will be 1 minus beta, okay? So these are simple Bernoulli probabilities, distributions. Now you can imagine that where do we get the values of alpha and beta, okay? They can be estimated during the learning process. We can, as I said earlier, that we can assume a prior distribution for alpha and beta, and while learning the tree and the genotypes itself, we can also learn alpha and beta, okay? So now we have to have another probabilistic model for the single cell read counts. So what are the read counts available to us? These are the read counts that are available to us. We have a reference count matrix and a variant count matrix. So what is this? Essentially, the reference count matrix is consisting of this kind of entries, Rij, where basically these are count values corresponding to positions in different cells, okay? And this is basically saying what is the number of reads consisting of or containing reference allele in that particular position in that particular cell, okay? So that is the reference count matrix. We have this variant count matrix, Vij, that is saying in the ith position in jth cell, what is the number of reads corresponding to the variant allele or mutated allele. So this is our observed data. This is really the observed data coming from the sequencing process, right? So now we know that this reference count matrix or the variant count matrix, this should be dependent upon the genotype after amplification, because that was the data generation process itself, right? So the probability of Rij, Vij given psi ij, so this is a probability, conditional probability distribution that we have to define. So these are, because these are count matrices, we can simply define them using binomial distribution. Okay. So what we are saying is that in the first scenario, if we say that psi ij is equal to 0, that is the library genotype is unmutated, right? Then mu0 is basically the probability of observing a variant allele, and this is nothing but corresponding to something like a sequencing error. So if we say that the library genotype is unmutated, okay, then what is the probability that you will see a variant allele? The probability is only coming because of the sequencing error. So mu0 is basically 0.001. This is the probability of sequencing error in let's say Illumina sequencing. Yes. Yes. So dropouts have already been modeled in the previous equation over here using this false negative rate. Okay. In the other scenario, if we are saying that yes, library genotype is 1, and that means we can model it again using binomial distribution, where Cij is nothing but the sum of Rij and Vij, right? That is the total count, this is the variant count, and mu1 is the success probability of observing a variant allele, which is nothing but 0.5, because if the library genotype is mutated, we should at least see, I mean, by on an average, we should see 50% of the reads to be mutated, okay? Because we are considering heterozygous mutations. So this is the single cell read count model. So now I think we have every component to define the total probability of our model. So this is how the log likelihood function actually looks like, okay? So look at this particular function. What do we have? This is our observed data. This is reference count. These are the variant counts, V. And then we have all these different parameters. G is basically the true genotype matrix, which we are trying to infer. T is basically the tree. M is mutation placement on the data, on the tree. Alpha and beta are the false positive and false negative rates. Okay. So it turns out that G is basically defined in terms of T and M. It's as simple as that, because let's say these are our cells, right? This is C1, let's say C2, C3, and so on. And if we place the mutation over here, then we know only C1 is mutated, all the others are unmutated. Similarly, if we place it here, then we know C3 is mutated, others are unmutated. We place it here, we know whatever cells are present over here, they are mutated, everything else is unmutated, right? So given the tree structure and the mutation placement vector, G is already determined. And that means once we have G, we do not require T or M, right? So that means this whole thing can be converted into something like this, that we know that given G, T and M are not required, because G is already defined in terms of T and M. And if we break down this term in terms of number of sites and number of cells, we'll get something like this. What is this? This is very simple, that basically we are having a log likelihood function, so that is the product is becoming a sum. So sum over the number of sites, sum over the number of cells, and inside each, what we are seeing inside the log is the probability of this two-step process, that given the true genotype, we are obtaining a library genotype, and from the library genotype, the sequencing data is being generated, right? So that is what we are seeing over here, that Rij, Vij, these are the read count data being generated from psi ij, and psi ij is generated from Gij and alpha and beta, okay? So now this whole formulation is giving us the total log likelihood of our observed data. Yes.
N is the number of cells.
No, no. This M you are talking about. Yeah, this M is the number of cells.
So if that is the sum over the number of cells, then is it assuming that each cell is independent? Because
No, this is not assuming each cell is independent. But why is it a sum over the number of cells? Each cell is not independent. Each cell is connected via the tree. But once you have the G defined, which is already taking into account T and M, then we can do this as a summation. Okay. So then this actually there are two M's. Maybe that's why you got a bit confused. Okay. So this M that I'm talking about, this M and this M are the same. This is the mutation placement vector, and this M is referring to the number of cells. Okay. So now we can define our maximum likelihood estimate solution that we want to find out the G, T, M, alpha and beta, or rather G is already going to be defined in terms of T, M, alpha, T and M, and alpha beta of course we have to infer, and then we can obtain this MLE solution wherever we are maximizing this whole log likelihood function. Okay. So how do we do it? We resort to this heuristic search process, so which is basically a hill climbing algorithm we are applying. Again, this particular scenario is very similar to what people have used in the field of phylogeny. So we start with a particular tree and mutation placement vector, and then we compute the likelihood. Then we make changes in the tree, and for each tree, we can actually find the most optimal placement of the mutations, right? By changing the placement of the mutations on the branches of the phylogeny, and that would give us different likelihood values. And by doing this iteratively, we can make changes in the tree topology, and we can find the optimal tree. Fine. How do we change this error rate and error rate parameters, that is alpha and beta? Again, what we can do that from the very existing data sets, we have sort of a rough estimate that what should be the value of alpha and beta, or in what range this alpha and beta should lie. We can start at those values. We can assume a normal distribution, and then we can sample from the normal distribution and see which values are actually improving the likelihood. Because we are looking for a point estimate, so ultimately we are going to end up with a point estimate for these kind of likelihood functions. Okay. So one, I would say, very interesting problems that we encounter in working with this kind of problems is that we do not have any real data with ground truth solution. So we'll have to simulate realistic data sets. So that's what we did. We did the benchmarking on simulated data, and we compared against two methods. So one of them is definitely sci-fi, which I said like earlier, that was the very first method which started working with or started treating these two problems together. So what you see here, and the other method is monovar, which again I think we developed back in the day. This was the very first mutation calling algorithm for single cell sequencing data, but it does not assume any phylogenetic structure or anything. So one thing you can clearly see that F1 score, again we are looking at F1 score as sort of our measure of accuracy. We want to, the higher the better. So when we are varying the number of mutations or number of cells, we see that for every scenario, sci-fi and phylovar, they are having almost similar accuracies. And of course, both of them are sort of better than this monovar, which is not utilizing phylogeny information. Okay. But at the same time, what we also see is that if we do look at the runtime, right, we see that phylovar is much faster than sci-fi. And as we are increasing the number of mutations, the growth is also kind of smaller as compared to sci-fi. Still, of course, it is still more than monovar, which is not comparing using any tree structure, but it still is much better than sci-fi. So that means now we have an algorithm which can scale with the number of sites. It has very good accuracy, And essentially with a practical scenario of, let's say, where we can have single cell whole genome sequencing data, we can run it without running into the issue of runtime, okay? And this plot over here is also showing you how accurate our estimate of the error rates are. So on the x-axis, we have the true allelic dropout rate. And on the y-axis, we have the estimated value of beta. And we see that these two are very much correlated, okay? So then we actually applied on some real cancer data sets. So this is a whole exome sequencing data, which is coming from a TNBC patients, triple negative breast cancer. This, I think, contained around 16 cells, 16 tumor cells along with some normal cells. So, so I think when we developed the method at that point, not many barcode datasets were available. Maybe now it should be possible. But then again, here we are looking at something slightly different. So for example, in order to have the kind of benchmarking that you are talking about, we have to have lineage barcode data as well as single cell whole genome sequencing data. So I don't think that kind of datasets are still available. So again, this is just to show you the application of Phylogeny on some real cancer data set. So what you see here, x-axis represent the cells, y-axis is basically genes. So initially we filtered around 3,000 candidate loci. And after we performed this joint analysis, we actually found that out of these around 652 loci were actually containing SNVs with high or moderate functional effects, okay? And among these loci, 550 of them were also supported by bulk data. So now you might ask why we are looking at bulk data, because one thing is, the scenario is that yes, if the mutation is also present in the bulk data, that means we are also reliably able to detect the mutations that are present in bulk data. Plus we are also finding some new mutations, right? And for this particular data set, again, the result of Phylogeny and sci-fi was kind of similar, the genotype matrix, if you look at it, even though sci-fi's result was little bit noisier as compared to this one. But what is important is that Phylogeny was 7 times faster than sci-fi. So in a practical scenario, this became much more applicable, okay? So from here, again, I'm going to switch a little bit towards another method, which is currently we are developing in the lab. And we want to highlight some of the things that were problematic, let's say with this whole literature of Phylogeny and sci-fi and so on. So what is the scenario? So we talked about infinite sites model. That we said, okay, there is mutations can occur at most once, and that's it. We cannot allow same site to be mutated more than once. That is not true. So there have been a lot of studies where people looked at different cancers, and they actually saw the signatures of infinite sites model being violated. So this is one such data set where same gene was mutated parallelly in different branches of the phylogeny, and even more violations were reported by different single cell sequencing data sets. That either they looked at, basically either they reported loss of earlier mutations, in some cases they reported parallel mutations, in some cases convergent evolution was reported. So that means it is not very realistic to use infinite size model, right? And what are the events that could actually happen in a cell lineage tree? Well, mutation, heterozygous mutation is one thing, but there are a lot of different events which are actually violations of infinite sites model. So what are these? There can be parallel mutation. So what you see here, this diamond structure is basically occurring twice in the tree. So it's happening on this branch and again happening on this branch. So this is an event of parallel mutation. there can be mutation loss events. So let's say this purple mutation actually happened initially over here, and then on this particular branch, somehow this got lost because there was basically a long deletion that happened, right? So this is a scenario of mutation loss. And this is actually quite common because we know aneuploidy is there in happening in many human cancers, or even large scale deletions at different levels, maybe kilobase or megabase level deletions happen. So somehow your SNV signature can get lost, right, over time. And then you can have some hemizygous mutation also occurring. So that means already there was a deleted region of the chromosome, and maybe on the other allele, mutation is being acquired. So all these different events can actually happen during the tumor evolutionary process. So we want to model this kind of events. So where beyond infinite size model, we want to go for something like finite size model of evolution. So where we can really say that yes, we are not only allowing heterozygous mutation, which are only occurring once during the evolutionary process, but there can be parallel mutation and there can be mutation loss as well, right? And the second thing that actually happened in, I would say, last 5 years is that this company, Mission Bio, came up with a machine called Tapestry. So what it can do, it can really increase the number of cells that are sequenced. So they are using targeted amplicon sequencing, and they are using a lot of different target panels, set of genes that they are targeting. These gene panels are actually very well defined for certain cancer subtypes, but you can have your own custom gene panel also. And then you can have this very much targeted high throughput single cell sequencing data. And what do I really mean by high throughput is that if you look at the number of sequenced cells, so this is a study from 2021, they looked at 123 AML patients, they generated single cell data using this Tapestry, and you can see that the range is basically in thousands. So previous single cell DNA sequencing datasets, they were actually facing difficulty in even getting 100 cells. Now you can get to a dataset consisting of 5,000 cells, 10,000 cells easily. So that means now we have really large scale single cell sequencing dataset, but this time the scalability is on the number of cells rather than being on the number of sites. And this is basically one of the figures from the paper where they looked at, I think, from 123 patients, they looked at more than 700,000 cells, and they looked at the presence of different mutations. So these are mutations. So you can see this particular small scale is basically 10,000 cells. So you can imagine that now we can get really high throughput data sets. Yes? It is possible, but what is very believable is that when you generate more and more such data sets, because you have the numbers, right, and you are looking at specific genes, you are actually getting high quality data set, because the more number of cells you have, you are becoming, you are basically having a data set which is much closer to the actual population scenario. Because if you see, when from the tissue we are taking only 100 cells, we can only say, okay, these are 100 samples. But it might be the case that you are missing some rare populations. But in this case, you have much more chance of acquiring these rare cells as well. Okay, so the question is, given this kind of data set where we have very large number of cells, but very small number of sites, how do we change the existing methods? How do we adapt them for this kind of data sets, right? So to that end, we are currently developing this variant caller, again, phylogeny aware. So this is taking the similar route as Phylover, except what we are adding that we know that cells coming from a tumor, they should have some sort of cluster structure. So that means when we are sequencing more and more cells, they should be somewhat coming from different tumor subpopulations. And that means we should be able to cluster them also. So instead of looking at cell level lineages, we want to build cluster level lineages. So that means we start with, again, this kind of data set where we have sites versus cells. We can apply our initial filtering, which are somewhat easy to do, to arrive to a situation where we have number of sites corresponding to, let's say, 5,000 cells. Then what we can do, we can apply an initial clustering. Yes? Clustering based on the mutation profiles, yes. So what we can do, we can just use this data set, which is again the read count data. We can convert the read count data into, I would say, some sort of likelihood matrix, okay? And on the likelihood matrix, we can apply some clustering algorithm. Now, this is the initial clustering algorithm, or this gives us initial clustering. After we obtain the clusters, we are again going to apply this kind of phylogenetic techniques, right? And we are not only going to focus on just the infinite sites model, but rather we can now model mutation loss, parallel mutation as well, right? So both mutation loss and recurrence can be modeled on this kind of trees. So we have devised a method for that. And then we are again doing this likelihood based search of the whole tree space to find out the final clonal tree as well as the mutations present in this tree. You cannot get RNA sequencing, but you can get some proteins. So there are multi-omics data where I think they jointly profile the DNA and some proteins. It is there, but maybe not at this scale. There are
Not at single cell.
no single cell level is there, but no, maybe not 5,000 cells. There are assays for doing that. For example, there is one assay known as G&T, genomic and transcriptomic, but these have not become very popular. I think people still need to improve the protocols. Okay. So there are some things that are different. Again, we, for the sake of time, I will not be able to go into the details. So what we did change is the likelihood model of how we model the read count distribution. So here we are using a mixture of beta binomial distribution, and that is based on the observation that this is how the variant allele frequency actually looks like. So we see that the beta binomial mixture, which is denoted as the red curve, is modeling the data much better as compared to binomial or just beta binomial distribution, okay? And based on these beta binomial mixture model, we can define some parameters using which we can model our nucleotide distribution. And this really came from another science paper which looked at single cell data sets, particularly the read count distributions. So anyway, I'll just quickly jump onto the result a little bit. So again, here we generated simulated data sets with 2,000 cells, okay? Luckily for us, 2,000 cells, other methods still run, okay? But you can see in terms of F1 score, they are not very good. Phylogeny is still doing okay. They are making some mistakes with respect to F1 score, but this is 2,000 cells. And this is what the runtime actually looks like, okay? Sci-Fi N is basically the updated model of Sci-Fi. It came out in 2022. And we are comparing against Phylogeny. There is another method called Sieve, which does not even run more than 50 cells. So we could not get any result out of that. So you can see the large amount of difference in runtime, okay? And this is only 2,000 cells. When we went to 5,000 cells, the other methods, we let them run for 4 days, no result, nothing. Whereas we could find it within 20-30 minutes, okay? So then we wanted to apply it on some real tumor data sets. And this is some ongoing analysis we are still doing, but I'll just show you some results. So this is on a data set from a TNBC patient. Again, it is using something called MPT-seq, which is nothing but a variant of the tapestry machine that were basically developed in the lab of Nicholas Nevin at MD Anderson. So there were around 4,000 cells from this particular data set. And what you see over here is these colors are basically denoting the tumor subpopulations that we inferred from this data. What you see over here is the UMAP representation of the same, okay, the colors. So in the original paper, when they looked at this data set, they only saw these two populations, not even two populations. I think they said that they found only one tumor clone. But we actually saw some more distinct mutations that we were able to recover from this data set. And then we are also finding a little bit more heterogeneity from the data. And we see the closely related clones as well. So this is actually the tree structure, what the tree structure actually looks like. So we see there were a bunch of mutations. In fact, there were some clonal mutations, okay? After that, there were many subclonal mutations that were acquired, and then we are seeing 5 or 6 clones. And we even try to do functional impact analysis for this set of mutations. So particularly, we see that some of these clonal mutations and subclonal mutations which appeared early in the tree, they were also having high impact. Okay, yeah. So that pathway analysis we are currently doing, so I will not be able to answer you right away. But one thing that is for sure is that, again, as I said, that this kind of data sets uses a set of gene panels, right? So the set of genes are kind of fixed that you can probe into. But even within those set of genes, maybe we can find out certain differences across the different patients. So as I'm saying, like we have not done that analysis yet, but we are doing that right now. So what we also found out is, so there were 5 patients in this category, right, from this data set. And what we found out is that like for every patient, we were able to find new mutations with very high functional impact according to this CAD score. And there were certain mutations that were repeating also. So for example, this mutation in protein C receptor, okay? This protein C receptor mutation, we are finding it very interesting because it's reported in multiple patients. We found it in multiple patients, and all of them were sort of clonal mutations. And we found out that it has some implication, like at least the gene is associated with poor survival in certain TNBC patients. So we are looking more into that, like looking at the domain where the mutation is appearing and so on. Okay. So yeah, I'm done. I am not going to talk about all this methodology. I think that is going to be too much. So I'll just quickly come to the summary. So again, I think the brief takeaway from this talk is that we need this kind of scalable statistical methods, again, for the purpose of mutation calling or tumor phylogeny inference from single cell DNA sequencing dataset. And not just DNA sequencing dataset per se, but there are other omics datasets which are also emerging, right? So people are generating ATAC-seq data. People are developing even methods for RNA sequencing data, even though that is more noisy in terms of mutations. But we need more and more this kind of scalable statistical methods. And we need to have this kind of scalability with respect to number of cells for sure, because most of the data sets nowadays, they are in thousands or even millions, right? And one of the other things that we observed that having a finite size model of evolution models the data better, that is for sure. Not only us, but some other groups have also reported the same. And that means we have to somehow model the mutation loss and recurrence. And another interesting direction is to sort of jointly utilize the copy number information as well. So far we have been looking into single nucleotide mutations, but maybe copy number mutations can also be added to that. And essentially, these methods will have applicability for tumor heterogeneity and evolutionary analysis. So actually, right now, we are trying to reanalyze that 123 AML dataset, and we want to see what new things we can actually obtain from that. So with that, I would like to thank my lab members and my collaborators and the funding agencies. And we are always looking for motivated students to join our team, so feel free to write to me. I'll just write my email ID in the board in case you need to reach me. And thank you all for listening.